Okay, can you hear me?
Yes, miss.
Hello? Okay, what are we going to do today? From here until your Cambridge exams, we're going to solve past papers. So I'm going to solve today the paper 4 exam. 1 minute, I'll share my screen. Okay, Omar, we'll wait for a couple of minutes until others join, okay? Can you hear me? Hello?
Yes, miss.
Okay, just 2 more minutes. I'm online. 2 more minutes until others join before we start. Can you see my screen? Hello? Omar, can you see my screen?
Yes, miss, I can.
Okay, first I want to know how was the paper 4 exam?
Very easy.
Very easy. Actually, unfortunately, you did not do well. I'm not talking about you in specific. I'm talking about in general. Paper 4 was not, the grades were very poor. Grades for paper 2 and 6 were much better. Okay, now let's solve it together because you all need practice in paper 4. Let's look at the first question. Figure 1.1 shows several villi from an Indian, which is part of the small intestine. State the name of one other part of the small intestine. What did you write on the exam?
Duodenum.
Yes, it's the duodenum, which is the first part of the small intestine. Give me a second. Good. Okay, then you have a diagram. You have the lacteals and the goblet cells and the area enlarged here. You always look at the picture that is provided. Figure 1.2 shows the tip of the villi in more detail. You have a goblet cell here magnified, microvilli, epithelial cells. The epithelial cells of the villi absorb nutrients by diffusion and active transport. Describe how active transport differs from diffusion. And we have 3 marks. What did you write here, Omar?
Active transport needs energy from respiration, so it moves from low concentration to high concentration against the concentration gradient.
Very good. What else? We want 3 differences. It's 3 marks. What else does active transport need?
Semi-permeable membrane.
Yes, it needs carrier proteins in a living cell membrane. And the diffusion, does it need energy?
No.
No energy. And the molecules, they move from low to high or high to low?
High to low.
High to low. And does it need a cell membrane or no cell membrane?
No cell membrane.
No cell membrane and no carrier proteins. Now, most of you, like you did, you wrote two differences, not three. You said that in active transport, the molecules move from low to high against a concentration gradient, and it needs energy from respiration. Correct. But you missed a mark. Because look at the numbers. 3, it's out of 3 marks. So you need to write 3 differences. So you lost the mark here. You understand where the marks go? Okay. Now,
Yes.
the second question. Explain the importance of the microvilli. How many marks? 2 marks. So I want 2 keywords. What did you write here?
The microvilli increases the surface area for better absorption of nutrients.
Yes, these are 2 keywords. It increases surface area for the absorption of the nutrients. And you can say how the nutrients are absorbed by diffusion and active transport, which they just mentioned in the question above. C, goblet cells provide protection for the epithelial cells that line the intestine. State the name of the protective substance produced by the goblet cells. What do the goblet cells produce?
Mucus.
Mucus, very good. You all got that. Now, the other question starts with the word suggest. The moment you find a question starting with the word suggest, this means that it's out of syllabus. It's a new idea. It's a critical thinking question. Suggest a protective substance or why a protective substance is needed in the intestine. 2 marks. Give me 2 reasons.
To protect the walls of a small intestine from the from the basic juices.
The juices, are they basic or they're acidic?
Basic.
No, the stomach produces hydrochloric acid
Yes,
to kill
بس
the
بعدين goes
bacteria.
to the مش
Okay,
بعدها
but
في
we're
bile
talking
juice
about
secreted.
goblet cells provide protection for the epithelial cells that line the intestine. State the name of the protective substance. Okay, if you said that, like it will protect from acids, from change of pH in general.
Okay.
This is what you mean? Okay. So the mucus will protect the walls of the intestine from extremes of pH, whether from acidic or alkaline. Good. So the mucus will protect the walls. So first, they already told you that it is protective. So you will need to give 2 protective measures. It will protect the walls of the intestine from any change of pH, whether acidic of P or basic, but it's very basic in this one. And it will protect it also from what? Always look at the number of marks provided by the question. This is not enough. This will give you 1 mark out of 2. Think, this is the suggest question. So what do you have in the intestines that needs protection?
Protects the blood vessels.
No, think of something else. What do we have in the intestines? We have juices that contain enzymes and contain acids and bases and contains what else? So we need protection from what? You said from the bases or the acids. Okay, what else? We need protection from what? What is present that can cause harm?
Enzymes.
Exactly. These enzymes, they can digest the walls of the intestine. So we want protection from the enzymes. Okay, we also want protection from the bacteria. This mucus can trap the bacteria, just like it traps the bacteria in the respiratory system. Okay, so this mucus, they told you already that it is a protective substance. It provides protection. So your question or your answer should be, what does it protect you from? How many points do you need to write? You always go look at the number of the marks. You have 2 marks, then you need to write 2 things the mucus protects you from. Okay? A lot of you said it will protect you from bacteria, but you did not give me another form of protection. You also got 1 out of 2. Okay. Figure 1.1 shows a lacteal in the center of each villus. Describe the role of the lacteals. I want only one function because this is what you took. The lacteals have been canceled. We took it in detail 3 years before, but now it has been removed. You only know one function. What does it do?
Absorbs the glycerol.
Cholesterol and fatty
قصدي
acids.
absorb fatty acids.
Very good. But again, there is a continuation missing. So it will absorb the fatty acid and glycerol and return it to
Stop blood.
the blood. Correct. Just like the digested food, it is absorbed by the capillaries from the small intestine and it is returning back to the blood. So the 2 marks for 1 mark to mention that it will absorb the fatty acid and the glycerol, which are the large molecules that cannot be absorbed by the capillaries. And these large molecules, fatty acid and glycerol, will go back to the blood. Lots of like paper 4, the grades were not good. Not because you left it empty, even though some students left it empty, they did not study at all, but because always the answers are incomplete. You always give me incomplete answers. And then those incomplete marks, they add up, and then it causes a grade deduction in grade. Now, E, complete table 1.1 by identifying the level of organization for each structure. Choose your answers from the list. Each word or phrase may be used once, more than once, or not at all. Good. Now, the gallbladder. Who will tell me? This is an organ. Of course, the gallbladder is an organ. You don't know that? This is a problem. The endoplasmic reticulum.
Cell.
It's not a cell. No. It is the cell.
I
It's
mean
a
cell
separate cell.
structure.
Yes, it's a cell structure. Of course, the endoplasmic reticulum is an organelle. So it is a cell structure inside the cell. The epithelium. What is the epithelium? Is it
Tissue.
It's a tissue. In case you forgot, the tissues of the animal tissue, we took it in the first lesson, classification or organization of the organism. The tissues of an animal are semen, if you remember. Connective tissue, muscular tissue, epithelial tissue, and nervous tissue. Now the ileum, which is part of the digestive system,
Organ.
it's an organ. Okay. Now this is an easy question. Now this question is very important. Many fungi are decomposers that feed on dead plants. These fungi secrete enzymes to digest large molecules. Students made an extract from a species of fungus. The extract contained digestive enzymes. The students carried out an investigation to find out if amylase and pectinase, these 2 enzymes, and pectinase were present in the fungal extract. They made agar plates by filling Petri dishes with agar jelly containing either starch or pectin. They cut 4 holes of the same enzyme, of the same size, in the agar jelly in each Petri dish. And the holes in each Petri dish contained the same volume of hole A, 1% amylase, hole B, 1% pectinase, hole C, water, and hole D was the fungal extract. And the Petri dishes were kept at 27 degrees for 4 days. After this, a dye was poured into each dish to stain the areas where starch and pectin remained. Figure 1.3 shows the drawings of the stained agar in the Petri dishes, and the clear zones indicate the areas where no starch or pectin remained. So here it is talking about this experiment, and it described it in detail. They said that this Petri dish, the first Petri dish, contained starch. So this is starch. And in every hole, there was a different substance. A was 1% amylase. D was a fungus, a fungal extract. Okay, B was pectinase. B was 1% pectinase, and C, only water. And then they left it in this situation for a couple of hours or days, I don't remember. And then they looked at the clear zone. This is the clear zone. Now, what does this clear zone show? That the starch and pectin were digested by the enzyme or not?
يعني السترونج بتاع الستورشن عشان يعمل دايجيشن للأجار أو الجيلي موجود.
Very good. So I understand that you all know that this clear area, the clear zone, indicates there is either no starch or no pectin. Now, this question, a lot of you, maybe all of you, did not get the full mark on this question. Very few got the 5 out of 5. What are they asking about? State the conclusions that can be made about the enzymes in the fungal extract, okay, and give evidence from figure 1.3 to support your conclusion. So you want here to give a conclusion and evidence. So you go back to the question. Now, you all talked about, let me change color. You all talked about A and B. You all talked about A and B. You all talked about A and B. You never talked about, or A and D. You did not talk about B. You did not talk about, just give me a second. Let me erase it. What did you have to do simply in this question? You needed to talk about every Petri dish separately. So you needed to talk about the starch Petri dish and the pectin Petri dish. Okay, I want you to tell me, or to I want to ask you something. What is pectin? Because you should know what pectin is. We took pectin. Who remembers?
enzyme for fruit juice.
No, pectin is not an enzyme. It's pectinase, that's an enzyme. What is pectin itself? Pectin, we
Fungus.
took pectin in the lesson biotechnology. Who reminds me? Pectin, if you remember, this was the gluey material between the plant cells, and it was broken down by an enzyme called pectinase enzyme so that juice extraction was easier. Remember? We took it in biotechnology when we were studying how the enzymes help in biotechnology. So you already know what pectin is. And they already told you here what it is. No, maybe they did not tell you what it is, but they told you that there's an enzyme called pectinase. So now, what do you do in order to score the 5 marks in this question? You will talk about what happens in every Petri dish separately. So a perfect answer that was only made by mainly by only 1 or 2 students, you will talk about this Petri dish that contained starch, and then you will talk about this Petri dish that contained pectin, and you will talk about every disc, each one separately. So in the Petri dish that contained starch, A was 1% amylase. A was 1% amylase. And what did you say about A? Did it digest lots of starch or little starch? Compare it to D, for example. Can you tell me? Hello? Who will give me an answer? Can you hear me?
Yes.
Okay. Now, why is this out of 5 marks? Because you need to compare both Petri dishes, and you need to give me a comment or evidence from figure 1.3 from the first Petri dish, the one containing starch and the one containing pectin. I'm not going to waste any more time because I want to finish the whole exam. I'm going to, for example, if I want a perfect model answer, I'm going to talk about the Petri dish that contains starch. And I'll compare it with the Petri dish that contained pectin. Okay? And I'm going to talk about disc A, disc B, and disc C, and disc D. Disc A, in the first Petri dish here, that the one that contains starch, it contains 1% amylase. This 1% amylase did some digestion of starch. Can't you see that? Compare it with D. Did D contain more 1% amylase or less than 1% amylase? Look at the clear zone in A and D. Give me an answer. The amylase in A was more or less than the amylase in D?
More.
In A, why? Look at the clear zone. In A, is it smaller or bigger?
Yes.
Less. Okay, so A contained 1% amylase. and D contained the fungal extract. D contained the fungal extract. They both digested starch because I can see a clear zone around A and I can see a clear zone around D. But the clear zone around D is bigger than the clear zone around A. So this shows that D contains more than 1% amylase. Or you can say that D contains more amylase than A. Okay, so D contains more amylase than A. Why? Because the clear zone is wider. The clear zone, I can't write here, the clear zone is wider. A will have a smaller clear zone, smaller than D. This means that less starch was digested in A and more starch was digested in B, in D. Is it hard? Are you with me?
Yes, miss.
Okay, very good. Now, what will you say about B? B contains 1% pectinase. They told me that. I got the information from the question. Why is there no clear zone around B?
There was no pectin.
Yes. So you will say that there is no clear zone in B because there is no pectin. Now in C, it contains water. Why do we have C? C is for what?
Control.
Exactly. Okay, so at B, you will say there is 1% pectinase, but there is no clear zone because there's no pectin. At C, this is water. This is for control. This is for comparison. So here I talked about the first Petri dish that contains starch. Now we will talk about the second Petri dish that contains pectin, and you will divide it into 4 parts: A, B, C, and D. What will you say about A? A contains 1% amylase. Will you say, tell me, what did you say? In A, is there any clear zone?
No,
No, there is no clear zone. Why?
it doesn't contain any starch.
Exactly. A is 1% amylase. There is no clear zone around A because there is no starch. Finished. Now, what do you say about B? B contains 1% pectinase. There is a clear zone or no clear zone?
There is a clear zone.
There is a clear zone because some pectin was digested. Now, more clear zone than D, of course, you will compare B with D. There is a bigger clear zone in B or a smaller clear zone in B?
Bigger.
Bigger. So here I have a bigger clear zone. So this means that B contains more pectinase or less pectinase?
More.
More pectinase. Now let's look at D. D is the fungus. I can see a clear zone. So this means that some pectin was been digested, but the fungal extract has less pectinase than B. And what is C? We already said it before. This is the control. Okay? So this question is out of 5 marks. It's very easy. You will only simply compare both Petri dishes and you will compare every disk on its own. But all of you, maybe 95% of you, only talked about A and D and B here. That's it. So you lost a mark or you lost 2 marks when the evidence was not enough, the information is not enough. So you not only need to talk about the 2 Petri dishes, but you need to talk about everyone separately, every disk separately. You have to mention that C is water. Very little of you did not mention anything about C. A contains 1% amylase. A will break down the starch. Here there is no starch, that's why A did not break down anything and there was no clear zone. B is 1% pectinase. That's why B broke down some pectin in the second Petri dish and not the first one. D is the fungal extract. There is a big cleared zone in this Petri dish showing that there is a lot of amylase and there is more amylase in D than in A. Here, there is also some digestion of the pectin. This means that the fungal extract also contains some pectinase. C, in both situations, is the control. It only contains water. That is why there is no clear zone. That's it. You will compare this Petri dish With this petri dish, you will discuss every fungal extract, every disc on its own, and you will take the 5 marks. Okay, now you all know what you wrote on your exam. I hope you realize where you lost the marks here. Anyone has any questions? The investigation was repeated, but at 5 degrees instead of 27. So the temperature here was very cold. Predict the effect of the lower temperature on the results. What will the lower temperature do?
Slow down the enzymes.
What did you say? Slow down the enzymes. Yes, true. Now, when the enzymes, they slow down, what will this do to the clear zone? Bigger
That's
clear
known as
zones or smaller clear zones? Give me
smaller.
an answer. Smaller clear zones. Yes. All of you, also 95% of you told me that the enzymes will slow down. This is correct. And I gave you the grade for that. But when you go and look at the mark scheme, they want results. They want results. The results will mean smaller clear zone. Okay? If there is any clear zone around any disk, the clear zone will be small. Okay. Now, number 2, a scientist investigated sexual reproduction in flowering plants. Figure 1.2 shows the procedure for crossing 2 plants of the same species. You have to always look at the diagram. They did not here waste a whole page for no reason. There is definitely a reason. So the anthers are removed from flower 1, and then stigma ready to receive the pollen. They took the anthers from the male, and this is how they took the anthers here. And then they brushed it on the stigma. If you can see, the pollen has been transferred to the stigma to flower 1. So they have 2 flowers here. They took the anther from one flower, and they took a brush, and they took the paint brush used to pick up pollen from flower 2. And then the pollen was transferred to the stigma of flower 1. So here, what did they do? The anthers are removed from one of the flowers. They removed all the anthers. Obviously, they left only one anther. Okay, only one anther is left. And then they took the pollen from another flower and they brushed the pollen on the stigma of this flower. Okay? Now, why did they remove all the anthers? They only left one. What does the anther produce?
Pollen.
Pollen. Okay, so they took the pollen only from one of the anthers and they put it to the stigma. So this is artificial or natural selection?
Artificial.
Artificial. Okay, let's not waste time. And then a bag was placed around flower 1 tightly. So a bag was placed after they artificially took the pollen and they put it on the stigma of the flower, they covered it. Good. And then the seeds will develop in flower 1 after fertilization. The scientists collected the seeds and germinated them. The leaves and flowers of the offspring plants showed phenotypic variation as they were not all identical to the parent plants. The scientists then investigated the chromosomes of all the offspring plants and found that they had exactly the same number of chromosomes as the parent plant. Define the term chromosome. Now, there was the definition that is found in your notes, and I sent you another list of definitions that you have to memorize as it is. What is a chromosome? A lot of you gave me the definition of a gene, length of DNA that codes for a particular protein. This is not the definition of a chromosome. You need to memorize the definitions, whether you like them or you don't. A chromosome is thread-like structures of DNA, thread-like structures of DNA that contains genetic information in the form of genes. That contains genetic information in the form of genes. And it is a 2-mark definition. This is the first part, thread-like structures of DNA. This is 1 mark. That contains genetic information in the form of genes. This is the 2nd mark. So it's a definition that you have to memorize just as it is. Now, suggest why the scientists placed a bag around flower 1. Again, it's a suggest question, critical thinking. Why did they put a bag around this flower? You already gave me an
To
answer.
prevent
To prevent what?
pollination.
Yes, to prevent pollination from another flower. A lot of you said to prevent or to increase humidity. Now, that was a very funny answer. We're not doing here an investigation on photosynthesis. That person who said that I want to increase humidity did not even read the question. So we want to prevent pollination from another flower. To prevent pollination. We already did artificial pollination. We don't want any external pollination. Okay, if the Zoom cut off, because it will cut off on its own, we will wait for 10 minutes and then go back. Explain how sexual reproduction results in variation that the scientists discovered in the offspring plants. Now we took this sexual reproduction. How does it cause variation? I will not waste time asking you. Remember the MMRR that we took in adaptation and variation? We said sexual reproduction, with the best advantage of sexual reproduction is that it causes variation. And what are the causes of variation? MMRR. This question is out of 2 marks, so you needed to give me 2 out of those 4. The first M is because of meiosis. The combination of alleles in meiosis causes genetic variation. The second M is what? Who remembers?
Mutation.
Correct. Mutation, a change in the base sequence of the DNA. And then you have 2 R's. Both of them are random. They're both very easy to remember. The first random is random what?
Fertilization and breeding.
Very good. Random fertilization. I don't know which sperm will fuse with which egg. That's in the case of humans. In the case of plants, I don't know which pollen will fuse with which ovule. And random mating. I don't know which male will fuse or will fertilize or will copulate with which female. So sexual reproduction causes variation. And we said before that this objective was a new one added to the new syllabus, but you ignored it. Any question asking you about sexual reproduction and its causes for variation, you have to give the MMRR points. Mutation, meiosis, random fertilization, random mating. Okay, any 2 of those will give you the 2 marks. Again, 90% of you said meiosis only. You only mentioned meiosis and defined it. But that was not enough because they wanted 2 of the reasons of variation. Now, the chromosome number of the offspring plant is the same as the chromosome number of the parent plant in this investigation. Explain how the chromosome number is maintained from one generation to the next. Now they're talking about this specific investigation, sexual reproduction. So this reproduction here, so they're talking about meiosis or mitosis. When they transferred the pollen to the stigma of another flower. This was meiosis or mitosis?
Meiosis.
Meiosis. So how does the chromosome number remain maintained from one generation to the next? What will you write here?
The duplication of chromosomes before meiosis.
Okay, there is also something here that you should give me the answer. The pollen is haploid or diploid?
Haploid.
Of course. So here in this investigation, what did they do? They took the pollen from one flower and they put it on the stigma of another flower. And this pollen, if you remember, is going to grow a pollen tube and it's going to take the male nucleus to fuse with the female nucleus and the ovule. So the pollen plus the ovule here will give me a diploid zygote. So what happened is that the pollen is haploid and the ovule is also haploid. And they will fuse together to give me a diploid zygote. So here you will mention, how is the chromosome number maintained? It is maintained because the gametes are haploid, because the pollen is haploid and the ovule is haploid. And when they fuse at fertilization, they will give me a diploid zygote and we will restore the number of chromosomes. Now this we said so many times, okay? Because if the gametes were not haploid, if the gametes were diploid, the chromosome number will be doubled. And this is something that we do not want. So you had to mention in this question the 2 keywords, haploid and diploid. You had to say that the gametes are haploid. And when they fuse at fertilization, we will restore the diploid number. or n plus n will give me 2n. Anything that indicates the haploid and the diploid numbers. I hope you wrote that. Okay, nothing here we did not take before. Okay. If you have any questions, stop me. Now, also this question, most of you got this question correct. The plant camellia has flowers that can be white or red or a mixture. So these are the phenotypes, white or red or mixture of these two colors. When red flowered plants are crossed with white flowered plants, all the offspring have flowers with petals that are a mix of red and white, both. So this is an example of what? When I have two phenotypes in one organism. This is what?
Incomplete dominance.
No, this is not incomplete. You will tell me it's
Codom-
incomplete. This is codominance. You will tell me it's incomplete if the red flower fuses with the white flower to give me a pink flower, a third phenotype altogether. But this is not a third phenotype altogether. This is both phenotypes. So this is definitely codominance. Okay? Now the gene for the petal color in this plant is given as the symbol P. So P is the gene. And the allele for the white is P with a capital W. And the allele for the red is given with a P and a capital R. Table 2.1 shows the phenotypes of 3 different pairs of parents. Complete table 2.1 by giving all the possible genotypes of the offspring plants that could be produced by these parent plants. So when they give you an allele that is a capital letter, this means it's dominant or recessive, this allele. Capital letter.
Dominant.
Dominant, of course. Okay, now let's look at the genotype of the parents. And genotype means genotype, the genetic makeup of the gene in terms of the alleles present, not the phenotype. If the parents, both of them are red parent and red parent. What are the genotypes of both parents? Any parent that is red, what will be the only possible genotype? We said red is dominant and also white is dominant. But this parent is all red. So what's the genotype of the parent? I'm waiting.
R capital.
Yes. Only one R capital? Is the genotype only one
No,
allele?
two.
Yes.
Two.
So you can either write it like this was accepted, or the gene P with R above, that would be better, P with R above. So both parents were homozygous dominant. Now, the parents that are white, the other parent, no, one parent is white and the other parent is red. What will be the genotype of the offspring if I have a white parent and a red parent? What did you write on the exam? Give me an answer.
PRPW.
Yes. Now, white parent, this is also, we know that white is dominant. So the white parent is PP, W capital, or simply W capital, W capital. A red parent is P, red capital. And P is the gene. And this is how they told us. I did not make it up. Or it is R capital. So if you have a white parent crossed with a red parent, it will look like this exactly, because this is the space provided to carry out your work. This is a white parent crossed with a red parent. All the offspring will be heterozygous. All the offspring will be RW, will be codominant. You can write it like this, or you can write it EP, R capital, and W capital. All the offspring will have this genotype. If you wrote this answer, because some of you did this, I don't know why, this is wrong, because this is recessive. Okay, and they told us that the red is dominant. So the answer here will be P, R capital, W capital, or R, W. Now, petals that are both red and white, and petals that are both red and white. So the parents, what is the genotype of the parents to get the genotype of the offspring? Both parents are red and white. So what is the genotype of both parents? You just told me. Parents that are both red and white. Excuse me?
PW.
Yes. So this parent is RW. This parent is codominant, and this parent also is codominant. I will do a Punnett square, and I will see the genotype of the offsprings. This is R and W, and this is R and W. This child will be RR. This child will be heterozygous.